this is part two of our research presentations. We had another presentation a couple of weeks ago, and I did give an overview of the Science Cluster program at that time, but just in case you had missed it, the Organic Science Cluster program is the national science initiative of organic agriculture in the country, and it is funded by Agriculture and Agri-Food Canada. The Science Cluster is part of a larger program where all of the major commodity groups, they all have their own science clusters, and so in the current, we just are wrapping up our last cluster, which is a 5-year program, and I think there was 15 different clusters in that program. The program, the organic cluster itself, was valued at about $10.5 million, and 75% of that funding was coming from the federal government and 25% from industry partners. We would not be able to leverage the government funding without the support of industry partners, and so it's really, really important that we recognize the industry partners and that worked on this project with the researchers. And this included not everyone from farmers up to commodity groups that contribute funding to the organic research. So if we want to have research like this continue, we need to have the industry support to leverage that government money. The Science Cluster program that we're presenting on today is the second Science Cluster that we've had. It ran from 2013 to 2018, and it actually just wrapped up here at the end of March. We have submitted a proposal for another Science Cluster program to Agriculture and Agri-Food Canada, and we're hoping to hear sometime within the next month whether or not that proposal has been accepted. Next, we'll move on to Dr. Chantal Hamel. I'm assuming that she's going to be presenting today, and Dr. Hamel is a native of Quebec City and obtained her PhD from the Department of Plant Science of McGill University. And then she carried out her postdoctoral studies at the University of Montreal. Her research concerns the efficiency of nutrient cycling and the continuum between the plants and the soil, and has generated over 150 co-authored publications. In the last decade, she's had several publications concerning the influence of crop management on soil biology, mainly on the fungi, and in particular on the mycorrhizal fungi. So today she's going to be speaking on the topic of effects of mycorrhizal inoculation, rock phosphate and compost manure, and Pulse-Flax Rotations. And so welcome, Dr. Hamel.
Very much. I hope you can hear me well. I'm on the phone.
Yes, you're fine. Yep.
Okay, good. So I must say right away that I'm the one filling forms here, and the one who really did this work and the presentation, Dr. Yunliang Li, who is sitting right beside me, checking me in case I'm making mistakes. In the first place, I should not forget to thank the sponsors who made this research possible. They are the Saskatchewan Organic Directorate, Premier Tech, and the International Plant Nutrition Institute. I would also like to thank the organizer of this event to help us share our. Thank you all for your attention with this presentation. This morning, what we will present is results on the effect of mycorrhizal inoculation and 2 organic certified sources of phosphorus in Pulse-Flax Rotations. Next slide. For those who don't know, avascular mycorrhizal fungi, that we also call MS, are symbiotic fungi that help plants to take up phosphorus and other nutrients from the soil. And this is interesting for organic grain farmers, especially in the prairie, because the soil they have is often low in phosphorus. We also saw in previous work, inoculation with AMF could increase yield by 30% on an organic farm. We had 2 organic farms. One was benefited from inoculation, whereas the other didn't. So we can see sometimes inoculation can have effect, but sometimes cannot have it. So really the purpose of this study was to test some tools that might be useful to organic grain producer in the prairie. Next slide. We tested in this study if finely ground rock phosphate amended with humic acid could increase the level of available soil phosphorus and plant production, if composted manure can increase the level of available soil phosphorus and plant production, and if inoculation with AMF, the fungi, can help plants extract phosphorus from the soil. The next slide shows how the experiment was organized. So again, we were using a 2-year rotation. The first year was 2015. We had 2 sites, in Swift Current and in Beaver Lodge. In Swift Current, the pulse was lentil, and in Beaver Lodge, it was pea. And in the second year, flax was grown everywhere. The treatment, we had two types of called here factor 1 and factor 2. Factor 1 was inoculation with mycorrhizal fungi. In the first year, half the plots were inoculated with the commercial inoculant. And in the second year, half the plots that were non-inoculated were inoculated, and half the plots that were inoculated again. The factor 2 was fertilization, so we had treatment control where we applied nothing, and the other treatment were one of 2 levels of rock phosphate and one of 2 levels of composted manure. So we had rock phosphate, either composted manure at 2 different rates. The fertilizer were applied in the first year, and in the second year we just look at the carryover effect of fertilization the previous year, because we think the farmer don't want to go each year get in trouble putting fertilizer that are supposed to have effects. Next slide show that this presentation has 2 parts. So the first part, we look at the effect of the treatment, that is inoculation and fertilization by Phosphorus on plant trait and soil properties. So the next slide shows a lot of numbers in this presentation. So you should focus on the red circles. And the tables are arranged always the same way. So on the left-hand side, we have the treatment, inoculation on top and fertilization below. So here we look at the first year of the current. We see no red circle on top of so no effect of inoculation in the first year in Swift Current. Below we can see a few red circles hitting the effect high rate of composted manure. Composted manure in Swift Current reduced lentil yield, but despite increasing the percentage of nitrogen and phosphorus in plant tissue. So this looks strange, but we can tell that in 2015 it was a dry year in Swift Current, So we think that the effect of compost made worse the effect of water shortage. A good thing, however, was that soil phosphorus in the fall was increased with this high rate of composted manure, and this is good because that was an objective of the experiment. We can also point out here no effect of rock phosphate. The next slide shows the effect that we found in Beaver Lodge in the first year of the rotation. We saw small effect of inoculation that was reducing the phosphorus level in grain. That's no good. That's not the effect we expect from inoculation. But we saw good effect of composted manure that increased the yield of P. P was seeded in Beaver Lodge. Increase in yield, mass production with composted manure, and this associated with increase in soil phosphorus. So that was good because this is what we wanted to achieve with composted manure. The next slide shows what happened to flax in the second year in swift current. So we had only a carryover effect of the high rate of composted manure on the percentage of phosphorus in flax grain, and also on the soil phosphorus level, which is good. This is what we wanted to do. We saw no effect of rock phosphate once more. In Beaver Lodge, we also had a carryover effect of the high rate of composted manure in the second year of the rotation. So flax yield and biomass were increased with composted manure, and this was associated with increase in the level of nitrogen in the plants that was measured at bloom. Strangely, we also had a negative effect of the low rate of rock phosphate on the same parameters. So the conclusion from this first part of the presentation are that AMF inoculation did not improve crop yield or plant nutrition. Composted manure increased crop productivity and soil phosphorus level, and rock phosphate had no positive effect. In part 2, we are going to see the effect of the treatments on the microbial community of the rhizosphere. We thought that if we are using inoculant, we don't want to negatively impact the community that are residing in the soil. So that was the rationale for looking further into this. We look at the community of mycorrhizal fungi residing in the soil naturally, the community of general fungi, and the community of bacteria. So overall, the largest influence on microbial communities in that experiment was that of the environment, and this is not unusual. In this slide, you should see, I'm not sure if I say next slide, but it's 3 panels, bacteria, fungi, and AMF, and you should have an animation to help see the grouping here. So if you click enter, you should see the animation moving. So what it shows is that in that cluster analysis, we have the location and year that are clustered together. So that indicates that the community of the bacteria and fungi are the same within site and year, but differ between site and year. For the mycorrhizal fungi, the influence of the year was less, and this community of mycorrhizal fungi varied mostly with sites. So if we look at the next slide, we see we don't have any red circles. So that indicates that the inoculation and the fertilization treatment that we applied did not significantly affect the overall structure of any community. If we look at the next slide, we see the picture of the bacterial community in the different treatments. We can see on the top of the sheet Swift Current in 2015, that is first year and second year, and below the community in Beaver Lodge, first year and second year. And in the next slide, we have the statistics associated with this figure. And we can see a red circle only in the second half of the result sheet. So that means that treatment effects were only seen in Beaver Lodge. And we could see that inoculation and fertilization could influence the abundance of certain bacterial phyla in year 1 and year 2 of the rotation. The next slide shows the community of fungi, again on top found in Swift Current in first year and second year of rotation, and below in Beaver Lodge, first year and second year of rotation. The next slide gives us the results. We see the statistics that detect treatment effect. Only in Swift Current, we see that 2 circles on the upper half of the result sheet. So 2 significant results obtained in Swift Current, no significant result in Beaver Lodge. So we could see that inoculation had a slight influence on the abundance of the Basidiomycota, so the group, a taxonomic group of fungi. So that was in year 1. And we could see also fertilization affecting slightly the same phyla in year 2 in a carryover effect. The next slide shows treatment effect on the level of diversity of the microbial community. Significant effects were found in the second year of the rotation on avascular mycorrhizal fungi and bacteria. And we could see that repeated inoculation with mycorrhizal fungi could reduce the diversity of resident mycorrhizal fungi and bacteria. Also, fertilization had a carryover effect. Composted manure increased the diversity of resident AMF, and low level of rock phosphate increased that of bacteria. In conclusion, for the second part, we could see that overall, the microbial community were driven by environmental conditions at the year-end site, but treatments had little influence. However, on diversity indices, we could see effect. We saw that repeated inoculation with AMF could reduce the diversity of the resident mycorrhizal fungi in the resident bacteria. We saw also that composted manure could increase the diversity of resident AMF, and low level of rock phosphate could increase the diversity of bacteria. At the level of the abundance of phyla, we found little treatment effects, but here I would like to point out that it doesn't mean that we don't have effect on specific species or genera, because some organisms may be impacted in spite that at the level of phyla we don't see. Maybe we have some species, for example, that are favored and others that are decreased in abundance, but we don't see in the overall picture at the phyla level. We are not quite finished digesting the data, and we will check that shortly. So that's all I had to show today, and I'd be happy to answer some questions if you have any.
Great, thank you very much, Chantal. We have time for 1 or 2 questions, so we'll open the lines.
Chantal, Steve Shirtliff here. Hey, what was the phosphorus level of the soils that you were going into? Were they quite low, or?
That's a good point. I should tell that these experiments were conducted in research farm, and we look for low phosphorus level to be like we've seen in many organic farms. In Swift Current, the soil test P was around 10, and in Beaver Lodge it was much higher, about 25 to 30.
Okay, thanks. Yeah, I was suspecting, like Beaver Lodge, I think they've had a lot of cattle. Have they had a lot of cattle manure go on that research farm over the years? I suspect they may have.
Oh, I don't know. Maybe something to check, but I'm not aware of this. But phosphorus level was high.
Okay, great. Thanks.
Okay, we have time for one more quick question.
Andy, it's Becky here. Just a reminder to everybody to put your phone on mute, and if you don't have a mute function on your phone, you can do *6.
If there's time for one more question.
Please.
It's Stuart McMillan. So would your results, I mean, from a farmer's perspective, it could imply that, great, don't inoculate, don't apply rock phosphate, don't apply manure. Just keep on doing what you're doing. Would you actually take your results to lead to that sort of producer recommendation?
No, I would tell, because we saw some increase with the inoculum, and we see also here some effect of inoculation. They are not very marked, but we see impact, and we know that inoculation works sometimes doesn't work, and we know that the environment is very important. So there may be some condition where this, because there's one strain of fungi, mycorrhizal fungi in the inoculum, so maybe that strain likes some condition but doesn't like others. However, we don't know what are these conditions. So before we know what these conditions are, the recommendation I can say is you try it on a small area and see if it works in your condition. However, for composted manure, if you have the chance of having access to composted manure, I think it's a very good amendment. In Swift Current, we were not lucky in the first year with that drought year, but we could see that it increased the soil phosphorus level, and we don't have to apply it every year. As we could see in this study, we had a residual effect in the second year, increase in soil phosphorus as shown by the soil test P. So I think it's a good source of phosphorus if you can have access to it. However, the access may be limited. It is bulky, so you cannot carry it for long distance in an economical way. But if you have animals on farm, not many people do. You may have a neighbor with a feedlot, then that's real nice. But if you can have access to composted manure, I would highly recommend that you use it to increase the level of your soil if it is low.